This morning, Dr. Koob will discuss the effects of alcohol on the female brain. Thank you, Dr. Koob.
Thank you, Deidre. It's a pleasure to be here. I really want to thank Deidre Roach and Coralie Wetherington for organizing this. I mean, I know it was an enormous amount of work. I know Deidre didn't tell you how skeptical I was that she would be able to pull this off, but she did pull it off and in a major way, and we were very pleased. And kudos to both of you for this major effort. And I also want to thank the Women Drinking Pregnancy Work Group for their contribution to this. So, and Andrea Barthwell, I mean, you set me up perfectly, but what I haven't figured out is how I advance the slides. So here we go. So alcohol is a growing women's health issue. You heard about this. I'll just emphasize a couple of points that have struck me. Oh, and I want to mention that the reason Aaron White is on this slide, Aaron, where are you? Wave your hand. Come on. Is that he did all the work on this presentation, all right? And he did a yeoman's job because he had to do it in a very short time frame. But it is an area that I've been quite interested in, and I'll explain to you why as we move along. So over the last century, the gaps between males and females have narrowed for prevalence in drinking, total amount consumed, frequency, binge drinking, early onset drinking, having an alcohol use disorder, drunk driving, and self-reported consequences. These are all facts. In the last decade, differences have narrowed further for current drinking days per month, past use of DSM-4 alcohol abuse, and driving under the influence. And women are more likely now, we know, to experience blackouts, liver inflammation, brain atrophy, cognitive deficits, and certain cancers, as you just heard in elegant detail. In 2016, I was charged by Michael Gottesman, who's in the Office of the Director at NIH and directs all the intramural programs at NIH, to do a review on what we know about the neurobiology of sex differences in general, but that was way too much beyond my pay grade. So I decided with Jill Becker that we would do it on addiction, and the paper was published in Pharmacological Reviews last year. But one of the things that we noticed is a pattern that evolved from this. And if you take the 3 stages of the addiction cycle, which I will go through in a minute and as to why they're relevant, but binge intoxication, withdrawal negative affect, preoccupation anticipation, or the craving stage, you see definite patterns that overlap with the animal work. And that is that in humans, the females take less alcohol, although that's changing, but they escalate the use more rapidly, as you already heard from Andrea. In the withdrawal negative affect stage, it's pretty clear that in humans, females have a greater response than males. And in stress and anxiety-induced relapse, it's the same thing. And I hope to give you a little bit of neurobiological insight on why this may be the case in the next few slides. But the bottom line is that there's very little data, all right, at this point. So this was a review article, Sex Disparities in Substance Abuse Research, evaluating 23 years of structural neuroimaging studies. It's a really good article, but only out of 230 structural neuroimaging studies on substance use, only 79% included both sexes, but only 26% of those evaluated sex effects. And 85% of the studies that did stratify by sex reported different effects of substances on brain volumes, indicating the importance of sex comparisons. So we're just at the beginning, and I want to emphasize to you that when I joined NIH, the NIH in general and Francis Collins decided that sex as a biological variable had to transcend all elements of NIH's research. So those of you who don't do rat studies or mouse studies probably haven't noticed, because in human clinical studies, we've been including females all along. But now you are more or less required to include females as a biological variable when you do any research at NIH, whether it's a cell line derived from stem cells all the way to primate work. And so you're going to see, as a result of this, an enormous increase in our understanding of differences between males and females. So what I want to do is just very quickly, I want to talk about a couple of things relative to sex differences in the brain. So I am going to focus on the brain. So the motives for alcohol use, the reinforcement for alcohol use, the impact of alcohol on brain anatomy and function, and then withdrawal and relapse. So in the paper that Jill and I did, we identified 4 ways that males can be different from females and females can be different from males, okay? So there can be qualitative differences. Obviously, courtship behavior in mammals, who takes care of the babies, all of these are qualitative differences in behavior that are mediated presumably by the brain. There can be quantitative differences. As I know from rat studies, female rats, for example, take a lot more cocaine or drink a lot more alcohol than male rats. There can be population differences. It could be the percent of males that like a sweet solution versus the percent of females that like a sweet solution. But what I'm going to focus on today is the underlying mechanisms can differ. And they can differ in many different ways. They can be completely different circuits. Or I remember hearing a beautiful presentation just a couple years ago at the American College of Neuropsychopharmacology. You can actually have the same behavior manifested, but if you actually image the brain or look into the circuitry of that behavior, you'll find that females get to the same place as males, or males get to the same place as females, but they may be using different circuits or emphasizing different circuits or using a different transmitter within that circuit. So we're in for some really fascinating neurobiology, but then I'm a neurobiologist, so I think it's fascinating. But let me give you one quick example of the difference between male and female brains, and that is that females develop more interhemispheric communication. So it may be a little difficult to see on this slide, but the male communication within the cortex stays in the cortex. But the female communication from the cortex, as you can see in the bottom panels here, actually distribute themselves into subcortical areas. So you can read into this however you want. We're going to ultimately have to figure out what exactly this means about development. Those of you that have raised teenagers know that raising male teenagers and female teenagers is a different ball of wax. So you can think about this in that context. And there's widespread differences in morphology in adulthood that are being elucidated with brain imaging studies. And I just want to point out to you 2 that are maybe relevant, and keep that in the back of your mind. But in females, there's more gray matter in the medial prefrontal cortex. And in males, there's more gray matter in the anterior cingulate cortex. This is without drugs. And you can say, well, what on earth does that mean? Well, your medial prefrontal cortex is the brake on your reptile brain, okay? It's important for putting the brake on executive function, making sure that you don't make impulsive moves. So there may be something there, whereas the anterior cingulate is more involved in hedonic activities and releasing impulsive activity in your reptile brain. And it may be indeed that these 2 differences illustrate what I'm going to show you about the effects of alcohol on the brain. So now you're going to get a little Koob lesson on how the brain works in addiction. This actually comes from a paper that Nora Volkoff and I published just last year in, I think it's Lancet Psychiatry. It's an elaboration, a further elaboration of the neurocircuitry work that we've been summarizing and integrating over the last 10 years. And the argument is, and this was not handed down on Mount Olympus to Moses, and don't criticize I said that once. It's obviously an oxymoron. I said that once, and somebody actually wrote me a letter and said, an email and said I should take religious studies. But in any event, this is heuristic. That means it's something we use as a guide. So we know that addiction follows a binge stage. It's a big problem in alcohol use disorders. We know that when you're not binging, you're in withdrawal. And I want to emphasize to you, I'm talking about motivational withdrawal, not physical withdrawal. I don't think alcoholics drink because they get tremor, all right? And many of you heard me say that before. And then in between, there's a craving that develops. It can develop from many sources, but we call that the preoccupation anticipation stage. And you can make a pretty good argument that the bits of the brain that are involved in these stages are also involved in functional domains. So, for example, incentive salience is a binge intoxication domain. It's mediated largely by the basal ganglia, the blue bit there in the brain. And it involves reward and the linking of rewards to the environment, all right? And then the negative affect stage is generally, the kind of the downer stage where there's a reward deficit, but there's also, and you heard Andrea talk about this, a stress surfeit, an activation of stress systems. And that's mediated largely by amygdala, extended amygdala, a part of your brain that's critical for fear responses, fight or flight. And then the craving is frontal cortex, as I alluded to earlier. And there's a bit of the frontal cortex that can drive craving, and there's a bit of the frontal cortex that can inhibit craving, and those can be affected by drugs of abuse. And so you put this all together, you can come up with neurotransmitters that we know are involved, and so the questions are, do females start drinking for different reasons? Do the reinforcing effects of alcohol differ by sex? Does alcohol cause different brain adaptations in females? Do withdrawal symptoms occur? Are they different in males and females? And do the causes of relapse differ? And we're only at the beginning, only at the tip of the iceberg in trying to understand sex differences in these domains. But I can assure you that our portfolio at NIAAA, and I'm pretty sure NIDA's portfolio, are going to have lots of data coming in grants that are currently funded. So just a couple of quick things. We know that females show smaller increases in sensation seeking during adolescence than males. This is something we've known for a long time, that males are more likely to show sensation-seeking behavior. And the same goes for impulsivity in males. And this is, of course, a component of taking that risk of engaging in over-drinking or over-drug taking in the binge intoxication stage. And we also know that females may be more motivated to drink alcohol for negative reinforcement than males. And so this is a quote from the Kunsch paper where they say, the results from the largest drinking motive study conducted to date suggest that gender-specific prevention should take differences in the motivational pathways toward heavy drinking into account. That is, positive reinforcement seems to be more important for boys, and negative reinforcement for girls. Now, most of you are very well aware of the construct of positive reinforcement. So, go to SeaWorld in San Diego, watch the dolphin jump through a hoop, and watch carefully, and you'll see the trainer slip at a fish, okay? So that's positive reinforcement. But negative reinforcement is a neglected construct in psychology, and what it really refers to is the fact that you are trying to remove an aversive experience. So if you're having an anxiety attack and you're drinking to suppress that anxiety attack, which is more frequent in females than in males, that's negative reinforcement. You're trying to get rid of something aversive, okay? And some of you have heard this called, self-medication. That came out in an earlier talk. But one of the things we know is that drinking for negative reinforcement really, in my opinion, is often where you cross the line to what I would consider a moderate to severe alcohol use disorder, or what we used to call alcohol dependence, all right? And then anxiety and depression symptoms and alcohol use among adolescents is a study that was done, a cross-sectional study of Norwegian secondary school students, and increasing the severity of anxiety symptoms primarily associated with alcohol consumption measures among girls. And so there may be a link, as Andrea hinted at in her presentation, between what I would call these negative affective states and excessive drinking, and as we'll see, the telescoping that's associated with women's substance abuse. And so I just said this. I'll read the quote. We propose that males and females have different pathways of vulnerability to substance abuse. In adolescent boys, sensation seeking and impulsivity may drive drug and alcohol use, while stressful experiences and comorbid internalizing disorders may mediate substance use in adolescent girls. Now, those of you who are as old as I am remember a guy named Bob Cloninger. And actually, Cloninger proposed that there were two types of alcoholics at the time. He proposed a type 1 and a type 2. And the type 2 alcoholic was the top part of this quote, which would be impulse-seeking, sensation-seeking alcoholics. And the type 1 alcoholic would be the relief alcoholic. And that was it fits more the bottom or the female phenotype. And I might add that Cloninger predicted, even back then, 40 years ago, 50 years ago now, that in fact, his categorization of type 1 and type 2 were indeed loaded differentially for sex. So what are the exact differences in why females and males, the exact differences in why females and males drink are unclear, but it certainly does appear that alcohol affects the reward areas of the brain differentially in the two sexes. And this is a nice study done by John Crystal's group at Yale. I think it was John Crystal, which is the alcohol induces less dopamine release in females than males. And there's a linear relationship, you can see it there, between dopamine release and the subjective effects in men, but not in women. Dopamine is released in both males and females when you drink. This was equivalent about 3 drinks, as I recall from the study. And some of you are going to look at this and say, wait a minute, it looks like there's less dopamine on the alcohol side. And that is correct. It's actually what you're measuring, dopamine D2 receptor binding. So a decrease means that your endogenous dopamine has bound to the receptor and decreased that signal. All right, so low means high. Sorry about that, but it's a pet study. And even more intriguing is that the ventral striatum is larger than controls in binge drinking females, but smaller than controls in binge drinking males. And we don't really know what this means as of yet, but it is in the ventral striatum, and this is the basal ganglia, and this is, we know, the structure that mediates the rewarding effects of alcohol. You can interpret this in many different ways. One would be that females are protected here, but it could also mean that maybe this is the beginning stages of suppression of activity because of maybe an active neuroinflammatory mechanism. So, I think the mechanism for this remains to be determined, but there is a difference between males and females in this reward pathway. And then females show a smaller response of the amygdala to alcohol and corticosterone than males as well. Could this prompt more drinking? And this is a fairly complicated slide from my colleagues at the Scripps Research Institute, Marion Logrip, who's now an assistant professor at Indiana University. But there are differences. In the males, ethanol or corticosterone reduced the activity of these glutamatergic neurons in both the lateral and central nucleus of the amygdala. These are key structures in fight or flight and stress responses. But in females, they reduced this activation produced by glutamate less than in males. And so, it's the beginning of, and this was just acute binge equivalent administration of alcohol and measuring actually how these cells fire in the amygdala. So the central nucleus of the amygdala receives input from the basolateral amygdala, which receives input from the cortex. And so this is the way when you go into a freezing response when you're dodging a taxi at one of the circles here in Washington, that freezing response is the amygdala getting activated, and the pathway is through your eyes to your cortex to your basolateral amygdala to the central nucleus, which innervates the freezing response. And so, there may be differences in how males and females, how alcohol affects those responses. So one hypothesis that I ascribe to is that differences in neuroadaptation and stress circuitry could lead to differences in motivational withdrawal. And so the quote here is, importantly, there is evidence that while severity of alcohol withdrawal may not be substantially different between men and women with similar drinking histories, the qualitative nature of the withdrawal may be indeed divergent, suggesting that profound differences in neuroadaptation to alcohol dependence in men and women. So during withdrawal from other substances, perhaps alcohol too, stress reactivity and cortisol levels are higher in females than in males. And this has actually been borne out with a study on fear-potentiated startle results. This is a fairly complicated slide, but the top right-hand panel shows you that women show a much more dramatic cortisol response when they are exposed to a startle stimulus, which is a fear-potentiated startle stimulus. So where a cue has been paired with something that makes you jump, all right? And the extinction is also slower in females, and that's the bottom left-hand panel. So, there's hints, beginning to be hints, that the brain of females reacts differentially in the context of drugs of abuse during this withdrawal negative affect stage. And then we know that females exhibit more damage to the hippocampus, frontal lobes, and other areas following repeated chronic high-dose alcohol exposure than males. Could this be contributing to larger cognitive deficits in females? Is this part of the escalation? Is this part of the telescoping to a more severe alcohol use disorder? And it's a hypothesis, but again, there are thicker frontal cortex areas in binge drinking females. They're thinner than in binge drinking males. This is the red there is the frontal cortex that I've been talking about. Again, an intriguing observation. We're not exactly sure what it means at this point, but there is a difference between males and females in the third part of the brain and in the third domain, which is this craving domain. So what do we know? Just to summarize what I've told you, we know that females may be more motivated by negative reinforcement. Males may be more motivated via positive reinforcement. The reward system seems to be less responsive to alcohol in females and shows fewer neuroadaptations with repeated exposure. On contrast, the stress areas may be less affected by alcohol in females initially, but chronic use may lead to greater rebound increases in activity, may be related to cortisol. One of the little things that they don't teach in medical school is that if you have high chronic activation of cortisol, it drives the stress systems in your brain. And I don't have time to go into all that, but I'm going to repeat it. High chronic administration of cortisol, because of chronic stress, it would normally make you cushingoid, but there's a mechanism in our brains to shut off the cortisol release that's in the hypothalamus, and that works fine. But there's another mechanism in the amygdala that cortisol is driving the brain's stress responses, and it may indeed be that females are more sensitive to that. There may be more damage and inflammation in the female brain during withdrawal, and chronic drinking leads to thicker cortical areas in women but thinner in men. What we do not know, and there's a lot, is how these neuroadaptations in the neurocircuitry change over time versus males. And we don't know the implications yet, as Andrea said, for prevention and treatment, including medication development. But I think this is a critical area, as Andrea Barthwell said, particularly the medications development. I don't know whether you all know this, but Janine Clayton always uses this example when she talks about male-female differences. But if you are someone who takes Zolpidem, that's Ambien, okay, for jet lag, which is to help you sleep. It's a short-acting benzodiazepine-like drug that's very effective in getting you 5 hours of sleep, all right? But if you're a female, you metabolize it at half the rate as a male. So if you actually take 20 milligrams of Zolpidem because you're flying from Europe and want to get a good night's sleep before you land in Washington, you should not be driving your automobile when you get out of the airport. Because if you're a female, because you're going to metabolize that 20 milligrams a lot less than a male, and in a lot shorter time, less time. So, keep in mind there are differences. It's unbelievable when you think about it that these differences in medications for psychiatric disorder are never or very rarely considered, because they become critically important. And of course, it's an area that we're going to have to put a good bit of emphasis on at the NIH. So again, I want to thank you all for your attention. Special thanks to Aaron White and Antonio Nerona, Jennifer Hoban, and Trish Powell for helping me get these slides together. Our interest in sex differences in the brain is a work in progress. Like I said, if I was to give this talk next year, we'll probably have even more data, and the year after, even more data. So thank you very much. I'm right on time. I'm right on time.